Thanks again for the invitation and the kind introduction. I assume that you guys can see me. Is
Yes,
that
it's
correct?
true.
Yeah, because I just see my slides, so I have to, be creative in to see your responses to what I'm saying. Um, the plan for the next hour, as far as I'm concerned, is I'm going to tell you a little bit about what, in my opinion, is not going very well in science nowadays. So what is wrong? And then I will briefly discuss two big solutions to these problems. And then I will really focus into a tiny detail that I have been working on over the last 15 years intermittently. That is the part about the importance of addressing limitations to our own work and show some humility. Um, and then, if we still have time, I'll give an example of a little project that we did at the Amsterdam University of Applied Sciences, where we have an open science checklist. And we wondered whether people actually adhered to the advice coming from that checklist. And then hopefully we'll have a lively discussions with you and hopefully address all the questions that arose. So going back to 2009 and 2014 with a series of 5 papers that were actually quite influential in how I thought about medical science. These papers were all published in the major journal, The Lancet, and the idea was that they claimed that around 85% of all investment in biomedical research was being wasted due to 4 reasons. And the 4 reasons are listed here in the 4 pillars. And the first one is basically about relevance of questions. And already we see low priority questions being addressed. And if we also look in that pillar, we all see a bridge to open science, that clinicians and patients are not involved in setting the research agendas, which is assumed then to be a reason of the questions being not always relevant to patient care. The second pillar of waste is closest to the heart of a methodologist like myself, a clinical epidemiologist, because it's about the choice of inappropriate research designs and also the choice of inappropriate statistical analysis. So, for example, it's that many studies fail to take adequate steps to reduce biases. The third pillar is about publication. How do we report our results? Here is a bridge to what I will dive into later, talking about limitations. But here it's mostly about a heavy selection process, which distorts the scientific picture that we see. So there is, on the one hand, complete suppression of the publication of full articles, and then in the fourth pillar, unbiased and usable reports, question mark. It is also about not reporting planned outcomes. So the study in itself may be reported, but then some of the results and the outcomes that had been planned and were actually measured are then being suppressed. So it's all about selective outcome reporting, creating a lot of problems in the picture that we have of science. So they also had some rough estimates of the magnitude of the problem. And the idea is that even if you start with 100% relevant questions, you would lose half underway, lose another half for reasons of bad reporting, and then lose another half because some of the reports, even if they are reported, are unusable or have incomplete reporting of all outcomes. And of course, when I say incomplete reporting of outcomes, it is usually about this misconception about statistics that's underlying this, where people just decide not to publish things because they are statistically not significant. This is a huge problem in the biomedical, but also in the social sciences. So you may, I think, doubt whether this multiplication rule 150, 25, 12.5 is actually valid because some of these reasons may actually be correlated. So the 85% may be slightly overestimated. But even if it were 60%, it would still be pretty disastrous, I think. So here is another illustration of this idea of, on the left-hand side, you see a picture of all the studies that have been performed. So it's the universe of all studies in a certain domain, let's say a certain scientific question. There are around 100 studies being performed. And if you go all the way to the right-hand side, you see you see the picture of what we see. So you see that the red, the negative, the statistically insignificant results have almost disappeared. And you see also that the number of studies out in the open is much reduced. And so the green is now pretty dominant on the right-hand side, whereas in reality, if we look at the universe of studies on the left-hand side, we see that actually we see the reality. And the arrows on top tell you the mechanisms that cause this phenomenon where complete studies are not being published, outcomes are not being published. There is creative use of language and subgroup analysis and what have you to change some of the things that have been reported into a more positive flavor. And then the final mechanism is the overcitation of significant results later on. So the big solution to some of these problems were open science. So the UNESCO has this influential report in 2021 that brings together a lot of initiatives around under the umbrella term of open science. When I spoke to people about open science, let's say in the 1990s, it was usually librarians that were very upset about the fact that let's say citizens in the world had no access to scientific publications. The well-known paywall problem that is still not completely solved. Yesterday, for example, I tried to read a particular article in Science, but I first was invited to pay $30 to be able to read it. So this problem I think, has been the start of open science in the 1990s. I think it was a meeting in where was this meeting in some Eastern European. I think it was in I'm not sure, Sofia. There was a meeting on open science, on open access particularly. But now in 2021, the UNESCO has really widened the whole umbrella of topics that fall under open science. So we see open science infrastructures, which is connected to this idea of having data stewards, making sure that everything is stored correctly and accessibly to other people. We have open engagement of societal actors, which of course is a link to citizen science. And on the left-hand side, we have open dialogue with other knowledge systems, which some people also link to decolonization of science and being more open to other forms of knowledge, where you see one of these circles. I don't know whether you can read it. I can barely read it. It says indigenous peoples and marginalized scholars. So open science is really much wider than it started out in the 1990s. One of the big solutions to the problems I just described in the beginning had already been addressed during World War II by the sociologist Robert Merton. In 1942, he published a paper, The Normative Structure of Science, where he had brought his ideas about how science worked, but also idealistic how science should work under this acronym QDOS, where the C is for communality, which in normal language just means that all findings belong to everyone. So each scientific product should be accessible to everyone of the community, which means like every citizen. And the U is for universalism, by which he meant it is not about who says something or who has a scientific claim, but you have to focus on the claim itself. So it doesn't matter whether it's a poor person or a man or a woman or the skin color. It doesn't matter. You have to focus on content. And the D of kudos is disinterestedness, which basically says the scientists should go for truth and not for personal gain. And the final one, the OS, is not for open science, but it is organized skepticism, which means we have to suspend temporarily judgment and sit back, reflect on what the claim is. Maybe if you're a methodologist, really think through how the analysis has been done, whether the study design really matches to the scientific question being asked, and things like that. And on the picture, you see the collection of old Greek skeptics. So now I want to move on to this, like I promised, to the next part of my talk, which is really zooming in into what seems to be a tiny detail, but I think it's important. It is about mentioning and paying attention to limitations in our own work. Of course, we are probably trained to look at limitations in the work we read, but I think it is worthwhile to try and be more self-critical within ourselves, which is probably the hardest thing. But we can ask the question, how can we organize skepticism within our team or even in our institute? So John Ioannidis, which is probably one of the champions in research on research, he's currently co-director of the Metrics Institute at Stanford University, together with Stephen Goodman. He in 2007 already wrote a paper about limitations, mentioning limitations, and said, well, if there are problems in the work we do, and the methods and the validity, we need to know whether these errors actually matter. And he also stressed that it would be interesting to know the extent to which they matter, which also, of course, is already a bridge to what I'm going to discuss in about 10, 15 minutes, which is about sensitivity analysis. What I find striking is that in many articles that I read, people say, well, you have to interpret this with caution. But of course, it doesn't tell you anything. What does it mean to interpret things with caution? So I would be far more interested to see the extent to which different errors or different decisions have an impact on the result that is being presented in a research paper. He says that it's important to pay attention to limitations because being open about limitations can inform the better design of new studies. And finally, as a cause of all these hesitations to be more open about limitations in our work, he said that the journal's instructions to authors tend to emphasize more the novelty and the impact and significance of our work than the limitations. So for me, an historical moment was actually the decision by the major American journal, Annals of Internal Medicine, in 2004 to force authors in their journal to add a little paragraph in their structured abstract. This is also the abstract that then appears in PubMed for everyone to see. And that little addition to the structured abstract was a little section with 2 or 3 sentences about limitations, and you had to add these. And the editors would also see to it that you were actually addressing relevant limitations and not just little stuff. And you see here on the left-hand side, you see the Annals of Internal Medicine, a random paper 2005, where you see this limitations edition, and you see another random paper from 2002 where this is lacking. So I thought it was quite interesting that this editorial board decided to do this. And when we published in Annals of Internal Medicine, we were also subjected to this new rule. But I liked it, and so it set off. Yeah, there we go. It set off a series of articles that I did with great help of a linguist and expert in natural language processing. It's Dr. Halil Kılıçoğlu, who was then working at the National Library of Medicine in the US. He has now moved. And we did a series of papers that are all around this topic of finding in an automatic way sentences in the discussion section of papers, sentences that have to do with self-acknowledgement of limitations. So here are some of the titles of the work that we did: Automatic Recognition of Self-Acknowledged Limitations in the Clinical Research Literature. It's in the right bottom corner. There is one in the middle. Because once you have automated the detection of such sentences, it's also easy. In the middle, at the bottom, you have the title, The Impact of Peer Review on Discussion of Study Limitations and Strength of Claims in Randomized Trial Reports. It's a before-after study. So we downloaded all the papers from journals that are completely open about their process. So you can have the first manuscript submitted, all the between versions, and then actually the final version of the published paper. And what we did was we downloaded the first manuscript that had been submitted and compared it in terms of self-acknowledgement of limitations, fully automatic, with the eventual publication that was out there. And of course, the differences in these self-acknowledgement of limitations would tell you something about whether there was an effect of the editorial process, including the peer review. And of course, you can read the paper, of course, but the effects were not huge. There was some effect. So it told us that peer review and editorial handling, at least in these journals, did something. These were the BMC series journals. They are completely transparent on what they do. And of course, you see here a more challenging title: Is the future of peer review automated? I don't think so. But although this has been published before the rise of the large language models and general AI, general AI, but generative AI. But we don't claim that the future of peer review can be fully automated when we publish this. But we thought that tools that can automatically detect problems in research papers, and this goes beyond detecting limitations, but also detecting other flaws, cannot fully replace the peer reviewers, but it can help editors and peer reviewers. And of course, if you move it upstream, it can also highlight things that authors maybe have forgotten. And once highlighted, they can repair it before they submit their manuscript. This work that we did with a small team of people, with Mario Malichki, who's also at Metrics in Stanford nowadays as a postdoc, this work links to the claim that John Ioannidis did, that journals' instructions to authors do not really invite people to be more self-critical. So what we did here, we took 835 journals from from Scopus, from all the fields of science. So we had a stratified random sample of these 835 papers, stretching from the humanities to the health sciences to life sciences, physical sciences, social sciences, and the multidisciplinary sciences. And we looked at different topics that had to do with open science and transparency. But for sake of time, I will only limit myself to limitations. And we see that John Ioannidis in 2007, he had a point, because I think this was published in 2019, 2020. It was still the case that if you go to the very right-hand side, you see that overall only 9% of all the instructions to authors across all the scientific fields paid any attention to, emphasizing to authors that limitations are something to be discussed in a paper. And we can also see that the health sciences are at 22, and the life sciences are at 11. So that's of course slightly better than the humanities, but there's still a lot of room for improvement. So what is a limitation? I think there are maybe four types of limitations, and I'm curious to discuss with you in the final part of this morning uh, about your ideas. I thought that maybe there's four types of limitations, where the first type is determined by the research question. So, for example, if we ask, does smoking cause lung cancer in humans? We cannot do an experiment because of ethics. So we have to do what epidemiologists call a observational study. And because of the lack of randomization, it is inherently weaker um, than doing an experiment. So that is a limitation that is caused by, one might say, the research question. The second type, one might say, is a consequence of a design choice. Let's take an example from qualitative research that I am at the institute that I work. Increasingly, people choose to do qualitative research. So let's say people say choose to do a focus group study instead of personal interviews. A potential limitation is that the question is whether a shy person would be giving you the same information in a group compared to had you decided to do a personal interview instead of a focus group. You don't know that. A third type of limitation is a consequence of protocol violations. So suppose you do a drug trial or a drug study, and there are some patients that just take half the dose you intended them to take. That is something that is, it's a limitation to the intention of your study. And currently, there are statistical tools to quantify the magnitude of the problem, which harks back to what Ioannidis already said, that it would be interesting if there are limitations to see the extent of them. What is the magnitude of these limitations? So number three and four are actually we can actually tackle using statistics. The fourth type I see is a consequence of choice in data analysis. So a classic example would be that epidemiologists would like to adjust regression models, for example, for age and sex. But if the situation calls for more adjustment and you would stick to age and sex, that will be a limitation. But increasingly, there are tools now to look at the effect of the different choices that we make and to be far more transparent about what would happen to our results if we make other choices. So limitations are usually written down in the discussion section. So if we talk about limitations, we talk about the structure of discussion sections of our papers. So I've did an informal review of several opinion papers that talk about the structure of discussion sections, or people complaining that there's a lack of structure in our discussion sections. So usually people say, let's start with main findings, and then straight away start about talking about weaknesses and strength of the study. And then have a section on meaning of interpretation of the results in the light of the totality of related evidence, usually systematic reviews. And then closing off with implications for care and policy, if there are any. That's not always the case. And then talk about implications for further studies and, more the implications for within science. And some people say it's actually optional to talk about mechanisms. I don't know, if you're in the life sciences, mechanisms are probably more important. This is more about clinical research. And then even people say, well, there's no need to do conclusions. The reader can do it for himself. Usually the conclusions are highly subjective anyway. So the contextualization of our work is an important part of science, of scientific writing. I think it is very helpful to make explicit the ideal study design. So, for example, if in any cause-effect question, in any causal study, I think it is very useful to think about the ideal study design, which is very often a randomized trial. The experiment is the strongest research design to go into causality questions, but it's not always possible to do that, as we have already seen in the smoking example. But it's good to have the ideal study in mind because it sensitizes us to the limitations that are inherent in choosing or having to choose for another design. Then during the study, you guys probably used to that, have a logbook, log the protocol deviations, and then start when we come to the writing, structure the limitations subsection in a cause effect study. You could think of contrast achieved. If people take only half the dose, that will affect the contrast between the control group and the intervention group. And of course, here are the favorite other topics that epidemiologists are fond about: selection bias, confounding, etc. And then a final section within the subsection on limitations, pay some attention to generalizability, which is usually not about representativeness, but is about effect modification. And very often, my experience is you have to bargain hard with your coworkers because a lot of colleagues, they argue that being open about limitations is like giving away free ammunition to your peer reviewers. And why should you do that? Have to be idealistic. We move to a radical form of transparency and quantifying the limitation section. So we go beyond the idea that we just say, interpret my results with caution, and nobody has any idea what you mean. Already in 2008, Timothy Lash said, move the debate among stakeholders from the realm of qualitative criticism, which is often heavily influenced by politics and polemics, into the realm of quantitative bias analysis. And then 12 years later, this was radicalized by the idea that we can reduce, or we should reduce, the asymmetry of information between the analyst, that's us, scientists, and the readers, and more transparently show what estimates are possible. And that is the idea of what some people call multiverse analysis or specification curve analysis. And as we will see, other people have called it vibration of effects. Here is an example of the package BOBA in the statistical package R. We don't have time to go into the details, but the idea is that you write a protocol with all the different decision nodes that you can distinguish, and then run the analysis, take away maybe the analysis that don't converge or just produce complete nonsense, and it's called pruning of the model with poor quality models. Get rid of those, and then finally you make the inference. And then there is, of course, the challenge to because you produce a lot of results, so you have to visualize those results in a sensible way. In observational epidemiology, in observational clinical research, seldom do we have consensus on which variables to control for. And many people who've studied the replication crisis, I don't think we have mentioned this term so far, but the replication crisis is just a phenomenon that people have found that even papers published in Science, Nature cannot be replicated, and probably just not true. Has to do with too many degrees of freedom that we as investigators have in all these different decisions, and multiverse analysis would force us to calculate the complete collection of the results that actually materialize out of these different decisions that we can make. So here's a team of around John Ioannidis again. He called the multiverse analysis vibration of effects. To just give you an example, it starts with a huge database, publicly open database, the NHANES database in the US. And they studied, just as an example of this multiverse idea, the effect of vitamin D on mortality. So the idea was, let's look whether vitamin D in a particular dose can prevent premature mortality in humans. And they had this huge data set. They had 13 variables, all binary variables to adjust for. And it was not an experiment, so there's lots of degrees of freedom for the researcher to decide, will I correct for one covariable or for 13? If you do it for 13, you have 2 to the power 13 different models, which is over 8,000 models, and you get a complex graph to show all the results. Just go back to the bigger picture. These are the same results. If you go to the top left-hand corner, you see the regression model of the effect of vitamin D on mortality if you have a crude model with no adjustment for any of these 13 variables, and then you go down diagonally to the bottom right-hand corner if you correct for all the 13 variables, and then you see on the x-axis how the hazard ratio or the preventive effect of vitamin D changes. And of course, you can say, well, it's quite sensitive to these decisions because the hazard ratio, the preventive effect, changes from around a protective effect of around 35% to a protective effect, if you control for all the variables, of around 25%. But still, the good news also is that the protective effect seems to be there regardless of the data analytic decisions. But the magnitude is much clearer. Here's the final example. This is from economics. Do women get a mortgage more easily than men? The dotted line just below the 4, the coefficient on female was just under 4. That would be like the classic thing that you would read in such an article. But if you show all the results of, in this case, 256 possible analyses with different degrees of adjustment, you see there is a full distribution of effects of being female and the probability of getting a mortgage easier than a man. Here again, too, you see that most of the coefficients still are in favor of an effect of being female, but there are some analyses that are coming to the left of the zero effect and pointing towards the reverse effect and men having a higher probability of getting a mortgage. This is the final example about multiverse analysis, which is a radical form of sensitivity analysis. Let's see. I have how much time do I have left, Elisabeth?
Yeah, probably 5 more minutes if it's
5
okay.
minutes? Okay, I'll run you through the little study that we did last year at our institute to get an impression about the adherence to this open science checklist that we had developed in 2020. So in 2020, we were inspired by these 4 sources of inspiration for the checklist. It was the Dutch code, scientific integrity. There will be a new release in this year. The second source of inspiration was the Avoidable Research Waste literature that I started my talk with. The third inspiration source was the replication crisis literature. Most well known maybe is Brian Nosek of Open Science Framework, who did a lot of work on replication problems in the social sciences and psychology. And the final one was some literature on open science, like UNESCO citizen science, and the literature on fair archiving. And so from these sources, we got a list of 14 items. We used it as a checklist. It begins with a very general one: get an ORCID account, an ID for an ISBN for every scientist. We stressed the importance of justification of relevance of your project, the involvement of end users. I started my talk with the research waste pillar 1, where there was this idea that not involving clinicians and patients was a source of poor questions. Then maybe some people are surprised by reporting guidelines being so early in the checklist, but we think it is right to anticipate on reporting early in your project, especially for junior researchers. And of course, there are some more idealistic. Well, let's not talk about idealistic things, but things that usually funders now simply require, like a data management plan and GDPR privacy issues that are just basically the law. And then of course, there are some more idealistic open science related items like complete reporting and pre-registration of protocol and analysis plan. And we simply set out, oh yeah, here is a slide which shows you the same 14 items, but in a clickable PDF. And our researchers can click on these segments and then go to a what we call an open science research manual to get some background information on what justification actually means and how you can do it and how you can really live up to the item on this checklist. And so in this project, which was really a small project, but it was still interesting to do, the goal was to gather data on the 4-year trends in the extent to which our researchers embrace the 14 principles of what we're going to say, the 14 open science principles, responsible research. And if we find any trends, interesting to see whether we can find reasons. A little bit of methodology. We had 4 years that we covered. We had 6 core departments, and because we had limited personnel time, we had to be very efficient. So we thought, let's have a stratified random sample again, with for each lectorate for each year 1 project. It didn't really work out like that, but it was the plan. So already we run into a remark on the limitations. We don't have time to go into that. I'll just go straight to the results. I will only give you the example for adherence to data management plan, protocol, and reporting guidelines. And well, let's focus on the main analysis, which is always in the top left-hand corner. We did a little bit of sensitivity analysis. It's not a multiverse, but it's still a little bit, taking a different decision. But for the sake of time, we'll focus on the main analysis. On the x-axis is the flow of time from the early starting projects to the projects that were starting more towards 2023. And on the x-axis is the probability of adherence. So these data were mostly built on desk research, where we looked at some of the output, but the most important part was actually a 1-hour interview with the researchers on a project to discuss what they did on the 14 items. And they could score 0 if we thought there's no adherence. They could score 1 if we said you knew exactly what we meant. And we could do partial adherence if it was, there was some things were done, but it wasn't perfect. And on the y-axis, you see the probability of partial adherence or full adherence. So it's a bit of a mild interpretation, but what we can see already is that a data management plan, the adherence is pretty high. It's almost perfect across all the years. When we move to the protocol means that if you have a text that you use to get the money, let's say the research proposal text that goes to the funder, and you get the money, I think ideally in science you would rewrite that text and produce a protocol that's much more detailed about what you're actually going to do in your project. That is what we meant by a protocol. And we see that already it's a bit lower than the data management plan. And if we then move to reporting guidelines, which is like a slightly less well known, at least in our institute, we see that the adherence is at a lower level and there's still much room for improvement. The confidence intervals around these lines are pretty wide because, of course, it's only a small project. So basically, what we found is that the 7 of the 14 items that are required by funders, by the law, or by our institute, they have excellent adherence. It's just a must. But the other more idealistic items, there's a lot of room for improvement. So this is my last slide for discussion, trying to get a bit more practical, because that was what we said in the title of my talk. Let's try to be a bit practical in the end. I think having a checklist and checking the adherence to that checklist is pretty practical. Um, we don't have let's say, we are not in a position yet as a support and open science support desk to, um, let's say, to force people to take on board all the 14 items. It's still like a little bit of a negotiation. And so that's that. And the second thing is that I think team science um, would come a long way to work in a more responsible way. And with team science, what I mean is that we get rid of the separation between scientists and support staff, and really acknowledge that all the different expertises are necessary for good science. And so you would have more interactive team meetings, whereas in our institute, that is pretty separate. And people are really free to consult us, so they can also avoid us if they like, as a support desk. How can we organize skepticism? I think it's important to do that in teams, and it is the active thinking about that and active invitation of strangers early on in the project. Because my impression is that if you're in a team, especially when the team is hierarchical, you're all focused on getting, we're all interested in and enthusiastic about the same things, and it's quite easy to have like a tunnel vision, and you need really strangers that are not so interested in what you're trying to prove to point out the things that you are just simply overlooking. I try to talk about slow science a lot, doing a bit less output and making sure that the output that you produce is actually completely adherent to the open science principles of transparency and accountability. Education of these principles, and not only to juniors, but also to senior people, often forgotten. And then, of course, the main issue is align the incentive structure with open science working styles. And then, yeah, with that, I would like to end, and I'm very curious about your questions and hopefully a lively discussion. Thank you so much.